All right. Welcome to the chapter on sensory receptors. So for starters, we're going to need to discuss this idea of sensation. And sensation can either be conscious or subconscious. You don't have to be actively aware of something for a sensation to have arisen, right? So this is going to be conscious or subconscious awareness of changes, and these can be in the external or the internal environment, right? So this is your body's way of sort of monitoring what's going on, right? And so the nature of sensations, as well as the type of reaction that is going to be generated as a result of these sensations, does vary widely depending on a couple of different things. But the main thing is going to be the final destination of where that nerve impulse is going to be sent to within the central nervous system. So where that final destination is within the central nervous system is going to influence, how we perceive these things or how we understand these sensations. So sensory impulses that reach the spinal cord. These are going to serve as input for things like spinal reflexes, which we'll talk about in just in the spinal cord lecture. These are going to be a great example of this is going to be the stretch reflexes. Sensory impulses that make it into the lower brainstem, so a little bit higher up than the spinal cord. We've made it to the brainstem. We've actually made it to the brain. This is going to trigger a far more complex set of reflexes. These can be anything from changes in breathing rate, in heart rate, and blood pressure, etc. So a little bit more complicated there. But the big deal is what happens when this sensory impulse reaches the cerebral cortex. At that point, this is when we're going to become consciously aware of that sensory stimuli. If it doesn't reach our cerebral cortex here, then we're not going to be consciously aware of it. All right. When that stimuli reaches the cerebral cortex, we are now consciously aware of that sensory stimuli. And we're also able to perceive the location as well as the specific type of sensation that we're experiencing, whether it's touch, pain, hearing, taste, whatever the case may be. All right. So now that's something that is sensation. This is not to be confused with this idea of perception. And perception is going to be the conscious interpretation of sensation. That is an important distinction to understand there, all right? And this is going to be performed primarily at the level of the cerebral cortex. Now, we have no perception of some sensory information, and this is because it doesn't reach the cerebral cortex, all right? Normally, we don't have any great awareness, conscious awareness, no perception, right, of our blood pressure, but we still have stimulus there. We still have that sensation, all right? We're just not consciously aware of it because it's not reaching the cerebral cortex. Another great example of that, there we go, is blood vessels, right? So those nerve impulses that are monitoring the stretch and the pressure that's being put on those blood vessels, that is being, monitored constantly, and it's sending those nerve impulses, but those nerve impulses are only going to make it to the medulla oblongata. They're only making it to the brainstem, and they fall short of the cerebral cortex, and so we're not going to consciously perceive blood pressure. Now, as far as sensation is concerned, we can break these down into a couple of different ways. But for starters, each type of sensation is going to be called a sensory modality. This can be touch, pain, vision, hearing, whatever the case may be. This is a sensory modality. And it's important to understand that a sensory neuron is going to carry information for only one sensory modality. This is a big deal, all right? You're not going to, perceive smells or odorants with your eyeballs, with your photoreceptors. That'd be insane. It is a one-to-one. A sensory neuron is going to carry information for only one sensory modality. Now, these sensory modalities get grouped broadly into general senses and special senses. We'll have an entire lecture series, 3 lectures or so, on the special senses. So we're going to focus today mostly for this lecture on the general senses. The general senses can be broken down further into somatic versus visceral senses. Somatic senses are going to be things like tactile or thermal or proprioception. Where is your body in space? Right? Whereas visceral senses are going to be things like pressure, stretch, the various chemical interactions you have in the various fluids in your body, as well as nausea, hunger, and temperature. Right? The special senses, on the other hand, are going to be the 5 big ones. Again, we'll have a whole lecture on this. We've got smell, taste, vision, hearing, and equilibrium. But the different types of sensory receptors are going to be involved with different senses. So we have different types of receptors for lots and lots of different types of these different types of senses. Oops. So in order to process all of these sensations, we're going to need a sensory receptor, right? This is going to be either a specialized cell that is designed specifically for sensing this stimuli, or they can simply be dendrites of sensory neurons. It just kind of depends on the type of sense that we're talking about. And a given sensory receptor is going to respond very vigorously. It's going to respond strongly to one particular kind of stimulus, right? Which is any kind of change in the environment that can be active, that's going to be capable of activating whatever our chosen sensory receptor may be. Now, a sensory receptor is going to respond only just weakly, and a lot of times not at all, to all other types of stimuli, right? Because of this, right, we say that our sensory receptors are selective. They have this characteristic of selectivity, where they're going to respond strongly to their particular type of stimulus and just kind of weakly or not at all to anything else. So they have that selectivity going on. So we've seen this kind of signaling arc before, but in order for a sensation to arise, it's pretty much the same thing. We have to have a stimulus, right? Some sort of change in the internal or external environment, whatever the case may be. Some sort of stimulus that's able to initiate a nerve impulse by the nervous system. All right, so we have to have our stimulus present. All right. Now, once our stimulus is present, a sensory receptor or a sense organ, whatever the case may be, is going to pick up that stimulus and it's going to transduce it. This is an important sort of step where we're converting the information from some sort of physical stimuli to an electrical impulse. This is when we're converting that physical impulse, that physical stimulus, into our nerve impulse, our electrical impulse. And this is done via a generator potential. Now, once our generator potential has been created, right, we're going to spark up our nerve impulse. And so our nerve impulse is then going to be able to, is going to conduct along our neural pathway from its receptor to the brain, okay? And so that being said, this pathway here, this trip that it has to take, so along that sensory neuron, this is going to be conducted from the peripheral nervous system into the central nervous system. This is a first order neuron, okay? These guys are important. This is what's bringing things from the environment, right, from the peripheral nervous system into the spinal cord or into the brain, right? Now, once that happens, then we have to integrate it. We have to decide what to do with this stimulus this nerve impulse once it's made it to this central nervous system. So a particular region of our central nervous system is going to receive that input, and it's going to integrate the sensory nerve impulses. Conscious sensation or perception, right, is going to occur when integration is happening in the cerebral cortex. Again, we're not consciously aware of anything unless it reaches the cerebral cortex. So, what we're primarily going to be focusing on today are the different types of sensory receptors. So, there are several different sort of structural and functional ways that we can classify these sensory receptors. We can group them into a couple of different classes. The way that we group them primarily is going to be based on how they look under the microscope, so their microscopic structure. Where is that receptor located, all right? Or what's the origin of the stimulus that's causing them to activate? And also, what type of stimulus is being detected, all right? So, starting with the microscopic structures, we've got three main options for these, all right? For starters, we've got our free nerve endings, okay? This is just a bare naked, right, dendrite that is on this the sensory neuron, right? When I say it's bare, right, it means it's not encapsulated. It doesn't have any sort of connective tissue wrapping around it, right? So these are going to lack any sort of specialized structures that you can see under a microscope. So they're just pretty plain Jane, just these free dendrites hanging out, ready to detect some sort of stimuli. We see this kind of free nerve ending receptor for things like pain, temperature, tickle and itch, as well as some of our various touch sensations. All of these are going to be managed by our free nerve endings here. All right. We then have the microscopic structure. We then have the encapsulated nerve ending. All right. So we've still got our dendrites up in here, right? But now we've added this layer of connective tissue wrapped around it. All right. And so we see this with both somatic and visceral sensations, mainly things like pressure, vibration, and some of our other touch sensations. These are all going to make use of these encapsulated nerve endings. All right. Their dendrites are enclosed in this connective tissue capsule, all right, that's going to look very distinct under a microscope. Lamellated corpuscles are a great example of this. They look kind of like an onion, with all of its little layers when we slice them and put them up under the microscope. And then our final type of class of microscopic structures that we use for classification of our sensory neurons, or sensory receptors, is going to be a totally separate cell, right? Where we have our sensory neuron is going to synapse with an entirely separate cell, all right? So we've got this separate cell over here that is going to be responsible for binding to or responding to that stimuli. Once that happens, this is going to trigger the release of neurotransmitters, which will diffuse across that synaptic cleft, bind to those dendrites, and are going to spark up our action potential. This is going to propagate along that nerve impulse, or that sensory neuron. Gustatory receptor cells that we have, that make up your taste buds, as well as photoreceptor cells in the retina for your eye, for vision, are both really great examples of these. So this is a really pretty cool situation here, where we just have a totally separate cell that is responsible for ultimately sparking up our action potential here and having that propagate down our sensory neuron. So a sensory receptor is going to respond to a stimulus by generating a graded potential, all right? And this graded potential is going to be known as a receptor potential. And in sensory receptors that are free nerve endings or encapsulated nerve endings, if that receptor potential is large enough, right, to reach our threshold, okay, if it's big enough to reach the threshold, then we're going to spark up our action potential. Action potential, all right? Very good. And this is going to trigger one or more nerve impulses that are going to propagate along that axon for our sensory neuron. Right? And the nerve impulses are then going to propagate along that axon, making into the central nervous system. Because remember, these are first order neurons because they're going from the peripheral nervous system into the central nervous system. All right? Here. Now, in the case of our separate cells, all right, the receptor potential is going to be generated when we have binding of our stimulus to our specialized cell. This is going to be what's responsible for triggering that receptor potential, okay? This is in turn going to stimulate the release of our neurotransmitters. These are going to diffuse across our synaptic cleft and are going to bind to and produce a postsynaptic potential in our sensory neuron, in our first order sensory neuron, all right? This is going to be important, right? So our postsynaptic potential is our type of graded potential here. And so if that threshold is reached, right, at this point, then our postsynaptic potential is going to trigger one or more nerve impulses, which are going to propagate into the central nervous system. All right? So that is how we classify neurons based on their microscopic structure. Come on now. There we go. Oops. I'm having trouble here. Okay, so that's our first way that we classify our sensory receptors based on how they look under the microscope. Another way that we can classify them is based on the location of the receptor or the origin of where that stimuli that's activating them is coming from. We have three options here for this. We've got our exteroreceptors, interoreceptors, and then proprioceptors. Exteroreceptors are monitoring the external environment, right? External environment. It kind of makes sense. Right? They are going to have their receptors are going to be located, either at or near that external surface of the body. Right? And these are going to be sensitive to stimuli that are originating outside of the body and provide information, again, about your external environment. So exteroreceptors close to the external surface and monitor the external environment. Right? Sensations for hearing, vision, smell, taste, touch, pressure, vibration, temperature, pain, etc., are all going to make use of exteroreceptors. Interoreceptors, though, on the other hand, are also known as visceroceptors because they are going to monitor the viscera. These are typically going to be located in things like blood vessels, your various visceral organs, as well as muscles and within the actual nervous system itself. And they are there to monitor the internal environment. So interoreceptor monitoring the internal environment. Nerve impulses that are produced by interoreceptors are typically not going to be consciously perceived. All right. That being said, if we have a very strong stimuli that is being, this could potentially trigger a receptor as pain, a feeling of pain or pressure. Think like heart attack. You don't typically, super aware of your heart, your blood pressure, your heart beating. But when you have a heart attack, that registers as pain or pressure in the chest. And that's because of that very strong stimuli. Our third type of location classification that we have is called proprioceptors. And these are going to be located specifically within muscles, tendons, joints, and the inner ear. These exist to provide information about your body's position. Where is your body in relation to gravity? How long are your muscles, and what's the tension that's being applied to them? What's the position of your joints and the movement and the range of motion that you have for those joints? That is all going to be covered under these proprioceptors here. And then finally, last but not least, the final way that we can classify these different types of receptors is based on the type of stimulus that they detect. All right. So mechanoreceptors are going to be sensitive to mechanical stimulation. All right. This is going to be things like the deformation or the bending or stretching of cells. This is going to provide sensations for things like touch, pressure, vibration, as well as proprioception, as well as hearing and equilibrium, interestingly enough. So anytime we have something physically bending, right, or physically stretching, this is going to likely be detected by those mechanoreceptors. They're there to monitor. These are things that are going to monitor the stretch of your blood vessels or the stretch on your internal organs, etc. All right. Thermoreceptors, the name should maybe give it away, hopefully. These monitor temperature, thermo as in heat, so they monitor temperature. Nociceptors are important, and they're going to respond to painful stimuli. And this can come about as a result of either physical or chemical damage to the tissue. Photoreceptors are going to detect light, hence the name photoreceptor, are going to detect light, and they do this as light strikes the retina of that eye there. Chemoreceptors, chemo as in chemicals, are going to detect chemicals, things like tastins. When flavor and tastins bind to your taste buds in your mouth, this is going to be making use of these chemoreceptors. Same deal with smell with our nose here. We've got smell, as well as monitoring the various bodily fluids as well, to some degree. And then last but not least, we have our osmoreceptors. These are there to monitor the osmotic pressure in the body's fluids. Pretty straightforward. So with these sensory receptors, we also have this really kind of neat characteristic that we see with them, which is this idea of adaptation, right? Where a generator potential or a receptor potential is going to decrease in amplitude during a maintained and consistent stimulus. So the stimulus is still there, is still present in the same amount, but we're suddenly having a decrease in the frequency or the amplitude of these receptor potentials that are being generated. This, as a result, is going to cause the frequency of our nerve impulses in our sensory neurons to also decrease. So we still have the same amount of stimuli, right? But we are now having a decrease in the number of action potentials or nerve impulses that are able to be generated, that are being generated each time. Right? And so because of adaptation, our perception of sensations can fade or disappear, even though that stimulus may persist. A great way to think of this is if you go nose deaf a little bit, if say, maybe you live with, you've got stinky kids. I certainly do, right? Or a cat. A cat's a great example, right? You may not notice that you're living with a cat or with a stinky kid, but somebody else might walk into your home and be like, ooh, it smells like stinky kid, or ooh, it smells like cat. Because you've gone nose deaf, you've had this idea of this adaptation taking place, where that stimulus is still there for sure, but we've had this decrease in the number of action potentials that are being generated. So, excuse me, receptors vary though in how quickly they're going to adapt. And obviously we group them into rapidly or slowly adapting receptors. Rapidly adapting receptors are going to adapt very quickly. All right, they are going to be great for detecting changes in that stimulus, but not necessarily super important for monitoring the continued monitoring of that stimulus. So receptors that we have associated with pressure, touch, and smell are all going to be rapidly adapting receptors. Slowly adapting receptors, on the other hand, are going to adapt more slowly, hence the name, and are going to continue to trigger those nerve impulses as long as that stimulus is present. Now, slowly adapting receptors are going to monitor stimuli that is associated with pain, the body's position, as well as chemical composition in the blood. All of these things need to be continuously monitored, and this is why we have these slowly adapting receptors that are used for this. So here, in case you wanted just a quick summary, is a table looking at all the different classifications of the various sensory receptors for your studying delights. So moving on then to our somatic sensations. These are going to come about as a result of stimulation of our sensory receptors that are going to be embedded in either the skin or the subcutaneous layer. All right, we also find them in the mucous membrane of the mouth, vagina, and the anus. There we go. As well as we also find them in the skeletal muscles, tendons, and joints. Okay. Now these sensory receptors for somatic sensation are going to be distributed very unevenly. All right. And so this means that some parts of the body are going to have a ton of these sensory receptors, and other parts are going to have much less. If you were like me and grew up in the 90s and early 2000s, it used to be a thing in like middle school to go around and try and lick each other's elbow without the other person knowing. And this is because there are very few of these little sensory receptors on your elbow. And so we just, it was a wild time. But anyhow, so again, we have this uneven distribution of these sensory receptors. This leads to something called a sensory homunculus. I think I spelled that wrong, but that's okay. You get the point. So the areas that have this high density of somatic sensory receptors are going to be the tip of the tongue, the lips, and the fingertips. Now, somatic sensations that are going to arise from stimulating the skin surface are going to be cutaneous sensations, right? Because they are associated with the cutaneous layer of the tissue, right? And we have four main modalities for our sensory somatic sensation. We've got our tactile, thermal, pain, and proprioception, all right? So looking at each of these individually, starting with our tactile sensation. This is going to include touch, pressure, vibrations, and tickle and itch. So although we are able to just perceive these things as different, right? The sensations, right, we perceive them as separate and totally different sensations. They're going to arise by activation of some of those same receptors. Now, several types of encapsulated mechanoreceptors are going to be attached to these large diameter myelinated A-type fibers that are going to help to mediate sensations of touch, pressure, and vibration. Other tactile sensations, things like tickle and itch, are going to be detected by free nerve endings that are going to be attached to these small diameter unmyelinated C-type fibers. So the tactile receptors that we see in the skin or the subcutaneous layer are going to include things like corpuscles of touch, the hair root plexus, as well as type 1 and type 2 cutaneous mechanoreceptors, plus the good old lamellated corpuscle and our associated free nerve endings. So, looking at these. Starting with touch. This is going to be one of those rapidly adapting sort of sensations. All right? This is generally going to result from the stimulation of tactile receptors in either the skin or the subcutaneous layer, just a little bit below. Now, there are two main types of rapidly adapting touch receptors. We've got our corpuscles of touch, also known as Meissner's corpuscles, and we have the hair root plexus. All right? Now, our corpuscles of touch / Meissner's corpuscles. This is a potato-potato situation. They're the same thing. Be aware of both names. They are both used frequently now, or still. So these are going to be our touch receptors that are going to be located in the dermal papilla of our hairless skin. Now, each corpuscle is going to be sort of this kind of egg-shaped, we can see it right here, kind of an egg-shaped mass of dendrites that's going to be enclosed again by our encapsulated, our connective tissue capsule, because again, this is going to be an encapsulated nerve ending. Now, because these corpuscles of touch are going to be rapidly adapting receptors, they're going to generate nerve impulse when they're going to spark up that nerve impulse when they have the onset of stimuli, and then they're going to sort of fade, right? These are found in high densities in the fingertips the hands, the eyelids, the tip of the tongue, the lips, the nipples, the soles, the clitoris, and the tip of the penis. The hair root plexus are going to also be rapidly adapting receptors. These are going to be found on hairy skin, as they are associated with hair, obviously, and are basically just free nerve endings that are going to wrap around this end around these hair follicles. Right? Now, the hair root plexus works by detecting movements on the skin surface when that little hair gets disturbed, right? So when a bug, right, lands on you and disturbs our little hair, okay, this is going to generate our nerve impulse whenever those free nerve endings are being stimulated, sparking up our nerve impulse. So those are our rapidly adapting receptors for touch. We also have two slow adapting receptors for touch. We've got our type 1 and our type 2 cutaneous mechanoreceptors. Right? Our type 1 cutaneous mechanoreceptors are also known as tactile or Merkel discs. These are this kind of saucer-shaped situation that we've got going on here. This is going to be flattened free nerve endings that are going to make contact with our tactile epithelial cell, with our Merkel cells that we have chilling in the stratum basale. These are going to be abundant in places like the fingertips, the hands, the lips, as well as the external genitalia. And these receptors are going to respond to continuous touch, things like holding an object in your hand for any kind of extended period of time. Our type 2 cutaneous receptors, on the other hand, are also known as Ruffini's corpuscles. And these are going to be these kind of elongated, encapsulated receptors that are located down in the dermis, as well as in the subcutaneous layer. But we also find them in other tissues as well. These little guys are very sensitive to skin stretching. All right, and this is like going to be when you if you've gone and gotten a massage or whatever, and you feel that person kind of smooshing you into the table, stretching your skin out. This is going to be our type 2 cutaneous mechanoreceptors, or our Ruffini corpuscles, that are detecting that. So our next tactile sensation is going to be pressure. This is going to be similar to touch. This is going to be a sustained sensation that's felt over a larger area than what we see for touch, and it's going to occur as a result of deeper deformation of either the skin and or the subcutaneous layer. The receptors that we see for pressure that are going to contribute to the sensation of pressure are going to be type 1 and type 2 mechanoreceptors. And these receptors are going to be able to respond to a steady pressure stimulus. And this is because, again, they are very slowly adapting. We next have vibration. Sensations of vibration are going to result from this rapidly and repetitive sensory signals that are coming from these that are stimulating these tactile receptors. Receptors for vibration sensation are going to be our lamellated corpuscles and our corpuscles of touch. Now, our lamellated corpuscles are also known as Pacinian corpuscles, is what we see here. Looks kind of like an onion. So be aware, be familiar with both terms there because they are still both used. So our lamellated corpuscles or our Pacinian corpuscles, these are going to adapt rapidly and are going to consist of our little nerve endings surrounded by multiple layers of our connective tissue capsule, again giving them sort of the resemblance of kind of like a sliced onion. They are going to be found in the dermis, the subcutaneous layer, as well as other tissues in the body. Our Pacinian corpuscles are going to respond to high-frequency vibration. This is like when you're using a power drill or you've been out mowing the lawn or other electrical tools, whatever that case may be. This is going to be high-frequency vibrations. Our corpuscles of touch, on the other hand, are also going to detect vibrations, but they are going to respond to low-frequency vibrations. So an example of these vibrations is when you move your hand kind of over like a textured object, like a basket or a paneled door. Those are going to be our low-frequency vibrations. And then we have this idea, these weird ones, these tickle and itch. These ones are goofy and I kind of think they're kind of fun. So itch sensation is going to come about as a result of stimulation of our free nerve endings, right? And they're being stimulated by various chemicals. This is things like bradykinins, histamines, or antigens. Things like antigens like in mosquito saliva that are going to be injected when you bite. And these are typically going to cause some sort of local inflammatory response. Now, the neat thing is when we have an itch, we scratch it and it makes it feel better, right? The reason that this works is because when we scratch that itch, it is going to activate a pathway that is essentially going to block the transmission of that itch signal through the spinal cord so that it is never able to make it to the cerebral cortex. We're not becoming consciously aware of it. It's kind of cool. Then we have tickle. These are going to be more free nerve endings that we associate with mediating tickle sensation here. This is kind of a weird one because this sensation is going to arise only when someone else is touching you. You can't tickle yourself, right? So why is this happening? Well, this seems like similarly to when we have an itch that we scratch, right? This is going to cause activation of a pathway that ultimately blocks that transmission. Similarly, when you try and tickle yourself, you're providing additional stimuli that's activating a pathway that's going to block that tickle sensation from making it to the cerebellum. So when somebody else tickles you, you don't have this additional stimulus, right? And so this information isn't able to make it to the cerebellum, and you feel like you're being tickled. Pretty crazy. So tickle and itch. So that brings us then to the very interesting phenomena of phantom limb sensation, right? So this is when patients have had a limb amputated, and they can sometimes still experience sensations, things like itching or pressure or tingling, even pain in that limb as if it was still there. This is, like we said, is called phantom limb sensation. So although the limb has been removed, those severed nerve endings of our sensory axons are still going to be present in whatever's remaining of the stump, right? And so if these severed endings are going to be activated, then the cerebral cortex is going to interpret those sensations as coming from the actual sensory receptors that would have been in that non-existing limb, or our phantom limb there, yeah? So another possible explanation for our phantom limb sensation is going to be that the area that we have of the cerebral cortex that would have been responsible for receiving and integrating that sensory input there previously is no longer needed, and so it's going to undergo pretty extensive functional reorganization. This is going to allow it to respond to stimuli from other parts of the body, but this reorganization, this remodeling that we see of that cortical area is going to give rise to sort of our false sensory perceptions from those missing limbs. So moving on then to our thermal sensation, right? Thermal sensation is going to be detected by free nerve endings that are going to have a receptor field of about 1 millimeter in diameter, so quite small. And this is found on the surface of the skin. And there are two distinct thermal sensations. There's one for cold and there's one for warm, right? And these are actually going to be detected by two different receptors. Our cold receptors are going to be located in the stratum basale of our epidermis and are going to be attached to medium diameter myelinated A-type fibers. Although there are some of the small diameter unmyelinated C-type fibers as well. These are going to detect temperatures between 10 and 35 degrees Celsius, all right, to activate our cold receptors. Our warm receptors, on the other hand, are not going to be nearly as abundant as cold receptors and are going to be located deeper down. They're going to be found in the dermis and are going to be attached to small diameter unmyelinated C-type fibers. And these are going to be activated by temperatures that are between 30 and 45 degrees Celsius. Now, cold and warm receptors are both going to adapt rapidly. They're going to adapt rapidly at the onset of that stimulus, but are going to continue to generate nerve impulses just at a lower frequency, even though they have throughout that prolonged stimulus. This is why when you step into a hot shower, initially it feels so flaming hot, and then eventually it feels nice and cozy. This is why. It's because we have this adaptation. It's not a complete adaptation, though. Now, any temperature that's below 10 degrees Celsius or above 45 degrees Celsius is not necessarily going to be detected by our cold and our warm receptors. Instead, this is likely going to be detected by our pain receptors that are going to produce sort of a painful sensation, which we'll discuss in just a sec. So, moving on to pain sensation. So, pain sensation. This is going to be a very important sensation because it is going to provide us with information about anything that could be potentially damaging to our various tissues. This is also going to be useful as far as a diagnostic tool to help diagnose disease or injury. Pain receptors are known as nociceptors. These are free nerve endings that are going to be located in pretty much every tissue in your body. And adaptation is going to be minimal because it is important. You don't want to be like, oh, my broken leg feels better, let me walk on it. So very little adaptation is happening. So a couple of different types of pain. We've got fast pain and slow pain. All right. So fast pain is going to be an acute, sharp, or pricking sensation. Think like when you get stuck with a needle for like a shot. All right. This is going to be perceived within the first second. All right, within 1 second. And the pain felt, again, from a needle puncture or from a knife cut this is all fast pain. All right. This is not going to be felt in deeper tissues of the body. Slow pain, on the other hand, is going to be more like a chronic, like aching, think like toothache. This is a great example there of a slow pain there. And slow pain can occur in both the skin as well as in the deeper tissues, and even our internal organs are all going to be great examples, are all places that we could have slow pain. And again, toothache is a fantastic example of slow pain. So when you perceive the difference, you can kind of notice or perceive this difference that we have in the time of onset. When we talk about doing an injury or some sort of pain to a part of your body that's farther away from your brain, right? So think when you stub your toe on some piece of furniture when you get up to tinkle in the middle of the night, right? You're going to feel that real sharp sort of sensation that's your fast pain, and then that slower kind of aching sensation is going to set in. That's our slow pain. And so we can see that difference in our time of onset very noticeably between fast pain and slow pain because that sensory information has a long way to travel. So as far as types of pain, again, we also have our superficial somatic pain. This is going to arise from receptors in the skin. Our deep somatic pain is going to come from the skeletal muscles, the joints, tendons, and the fascia. Right? Visceral pain, on the other hand, is going to be when we stimulate pain sensors associated with the visceral organs. These are usually going to be present either in or just deep to the skin that actually overlies that stimulated organ. All right? Now, as far as localization of pain is concerned, fast pain is going to be very precisely localized, right, to that area of stimulus. So when somebody sticks you with a needle for a shot, you know exactly where that shot was given. Somatic slow pain, on the other hand, is also going to be well localized, but it's a bit more diffuse. Right? This is going to involve a larger area and is usually going to appear to come from a larger area of skin. This is why when you stub your toe, you know exactly which poor little baby piggy that you've just squashed. And then as that slow pain sets in, we kind of just, all your toes hurt because, again, we've got that sort of more diffuse sensation going on with our somatic slow pain. Now, in some instances, we're also going to have, specifically when we're talking about visceral slow pain, the affected area is going to be where the pain is felt. So if the pleural membrane around the lungs is inflamed, you're going to experience that pain in your chest. However, that is not always the case. There are times when we have something called referred pain. And so in many instances of visceral pain, the pain is going to be felt either in or just deep to the skin that overlies that stimulated organ, or it can be felt far from that stimulated organ. This distance here is going to be called referred pain. So in general, the visceral organs that are involved in the area to which the pain is going to be referred to are going to be served by the same segment of spinal cord, and this is how we get this referred pain. So for example, our sensory fibers that we have for the heart, as well as the skin that's superficial to the heart, as well as the skin along that medial aspect of the left arm, are all going to enter the spinal cord segments between T1 and T5. And this is why when you're having a heart attack, at least if you're a guy, you feel chest pain and pain along that left arm there. So this is because of this. It's because of this referred pain, because this is being served by the same region of the spinal cord. That last but not least, then we have to talk about our proprioceptor proprioceptive sensations. Say that 5 times fast. These receptors are known as proprioceptors, and they are going to be located in skeletal muscle, tendons, and in and around the joints. All right. Oh yeah, and we also have them in the internal ear. We'll get into that when we do special senses. All right. But these are going to relate nerve impulses regarding muscle tone, movement of the body parts, as well as body's position in air. This right here is the goat Simone Biles, and she clearly has a very well, has excellent kinesthetic senses here. So this awareness of the activity of muscles, as tendons, joints, etc., as well as balance and equilibrium, are going to be provided again by our proprioceptive or our kinesthetic senses. So old Simone here clearly has some like next level kinesthetic senses here. Proprioception is also going to allow for weight discrimination. This is going to be your ability to kind of assess and guesstimate how much an object weighs so that you can put forth the appropriate amount of effort when you go to pick it up. That being said, if you go to pick up your coffee cup and you don't have this weight discrimination and you use the same amount of effort that it requires to pick up a small child, this is going to be a bit of overkill, and you probably throw your coffee cup halfway across the room. So again, this is allowing you to determine how much muscle effort is necessary to perform whatever task is at hand. We have 3 types of proprioceptors. We've got our muscle spindles, we've got our tendon organs, and we've got our joint kinesthetic receptors. So, starting with our muscle spindles, okay? These things are going to be found within the actual skeletal muscle, and these are going to monitor changes in the actual length of that skeletal muscle, and they're going to play a role in those stretch reflexes. Muscle spindles are going to consist of several slowly adapting sensory nerves that are going to have their endings wrapped about 3 to 10 times around these specialized muscle fibers called intrafusal muscle fibers. The main function of our muscle spindles is to measure muscle length to prevent any kind of overstretching potentially. Tendon organs, on the other hand, are up next. These are also going to be slowly adapting receptors. And these are going to be located at the junction between the tendon and the muscle. So where all of this stuff comes together. So here's our tendon organ we've got going on. All right. This is, by initiating our tendon reflex, those tendon organs are going to be there to sort of help protect the tendons and the muscles that are associated with them from damage due to any sort of extra tension on that muscle. So each tendon organ is going to consist of a thin capsule of connective tissue that encloses a few of our tendon fascicles. And these are just these cute little bundles of collagen fibers. Now, penetrating that capsule are going to be one or more of our little sensory nerve endings that are going to be intertwined around the collagen fibers that we see for that tendon. When tension gets applied to that muscle, when we start pulling one way or the other, all right, the tendon organs are going to generate nerve impulses that are going to propagate along into the central nervous system, providing you with information about changes in that muscle tension. So the result of this is going to be the tendon reflex that is going to decrease muscle tension by causing that muscle to relax. And then last but not least, we have our joint kinesthetic receptors, all right? We got a couple of different types of joint kinesthetic receptors, but these are going to be present within or around the actual articular capsule of our synovial joints. We have 2 nerve endings. That got away from me. The type of receptors we see for this are our type 2 cutaneous mechanoreceptors and our free nerve endings. Our type 2 cutaneous mechanoreceptors are going to be in the capsules of the joints, and these are going to both respond to pressure. We're also going to have these small little lamellated corpuscles that are going to be in the actual connective tissue also outside that articular capsule, and these are going to help to respond to acceleration and deceleration of the joint during any kind of movement. And then the joint ligaments are also going to contain receptors that are very similar to tendon organs that are going to help to adjust reflex inhibition for the various adjacent muscles when any sort of excessive strain has been placed on a joint. And so, there we have it. Here for your studying delights is a summary of the receptors for somatic sensation. That is everything for now. Oh, actually, I take that back. One last thing that is important. We've mentioned our first order neurons before, right? Our somatic sensory pathway. We mentioned our first order neurons. This is where we're going to have impulses coming from our somatic receptors into the brainstem or the spinal cord. So this is where we're going from peripheral nervous system into the central nervous system, specifically the brainstem or the spinal cord, all right? This is our first order neuron, okay? Our second order neuron, though, is going to be where that impulse is going to go from the brainstem and the spinal cord, and it's going to make its way to the thalamus. So we're going from the brainstem / spinal cord into the thalamus. All right? Okay. And then, so that's our second order neurons. And then our third order neurons, right, are going to be neurons that are going to convey that impulse from the thalamus to the primary somatosensory area in the cortex on the same side. So this is going to be from the thalamus to the cortex. All right. That is the long and short of the somatic sensory pathway that you need to know for this particular class. All right, and that is all I have for you. Thanks for listening, and see you next time.